The overall goal of this procedure is to isolate broad-spectrum antivirals from chemical libraries using a combination of in vitro cell-based assays. This is accomplished by first selecting compounds that inhibit the in vitro replication of a recombinant strain of measles virus that encodes luciferase as a reporter. In parallel, the compound's toxicity is estimated using a luciferase-based viability assay. The combined phenotypic data from both assays is used to isolate hit molecules. The final steps are to retest the hit compounds for dose-response replication inhibition of measles and chikungunya virus and to measure in vitro toxicity more accurately. Ultimately, broad-spectrum antiviral compounds are identified and validated by the screen.
The main advantage of this technique over existing methods like standard virus replication assays based on cytopathic effects is that it uses luminescence as a readout and combines two highly divergent viruses expressing luciferase as a reporter. Demonstrating the procedure will be Olivier Helynck, a technician from Hélène Munier-Lehmann's team, and Marianne Lucas-Hourani, an engineer working with me in Frédéric Tangy's research unit.
Begin by warming to room temperature the 96-well mother plates containing 10 millimolar stock solutions of chemical compounds in DMSO. Transfer 5 microliters of each compound to new 96-well plates, already with 20 microliters of DMSO in each well, thus creating intermediate dilution plates.